here was when I was in my first or second year of grad school. And I'm almost certain it was in this very room. I took a seminar from Tom Plummer on hominid paleobiology, but I'm not. And then the other thing I remember is doing a graduate seminar that met in your living room. And I think that was the very first talk, here we go, where I actually outlined, I said what I think I'd really like to do for my graduate research is I think we can probably collect urine from chimps and look at hormone levels and so forth.
And I think I told you it was impossible.
So yeah, I don't know what percentage of people in that seminar actually go on to do exactly what they said they were going to, but I certainly have. So what I really want to do today is talk about a number of slightly separate but interrelated studies that we've done recently at Kanyawara on male mating effort or male mating strategies. And I want to start by talking about the relationship between testosterone and male mating effort, which I've talked to some of you already. I know you're interested in this. I want to think a little bit about, so that'll mostly deal with male relationships with other males. And then we'll get into male relationships with females. I'll talk a little bit about sexual coercion. And then I want to think about some recent data that we've looked at on the costs of male mating effort, seeing if we can quantify the bad things about being a dominant male if you're a chimpanzee. So all of these data that I'll present today are from our field site in Kibale National Park that I co-direct with Richard Wrangham. He started there in 1987. I've been working there since 1996. You can see that's down here in southwestern Uganda. Here's the park. You can see it's what ecologists would call a hard boundary, I guess. As soon as you get to the edge of the park, it runs up against agricultural fields and so forth. And our community is right on the border of the park here. So the famous Ngogo community, which now has 180 chimps, is right in the middle of the park here in really prime chimp habitat. We're on the edge. It's not so good, but we still have almost 60 chimpanzees, and we're one of the few long-term study sites that's just maintained that number. So we have more chimps there today than when I started, which you can't say for very many of the long-term chimp sites, most of which have had precipitous declines over the last couple decades. But the chimps there have been habituated since about 1993, and really since 1996 or 1997 we've had really high-quality data on aggression and male-male competition. We've switched recently to continuous all-day focal follows. We used to do scan samples and shorter kinds of focal follows. And then since 1998, we've regularly collected urine samples every day, just opportunistically. The best way is to find a willing undergraduate to hold a pole like this and catch a sample. So that has allowed us over the last few years to look at all kinds of things, including most of our studies are testosterone, cortisol, looking at stress physiology, looking at ovarian function in females, and I'll show you some more recent data on using C-peptide as a measure of energy balance. And I'm going to talk a little bit about some life history stuff today, so I'll show you some graphs of ages of the chimps. So I should just mention that the first studies at Kibale took place in the very early 80s. So some of the oldest individuals, their ages are going to be estimates. Anyone who's under about 30, we know very firmly what their age is. For the rest, it's a little bit of guesswork. You can sort of put them in order, rank order of who's likely older than others just by physical characteristics. So the old males, have the wrinkled faces and the bald patches and the thinning hair. They don't have as much of the sort of glossy sheen to the hair that you see in the young males. You can look at the teeth and get an idea of the tooth wear with this kind of photo. So the very, very oldest individuals who are in the data sets I'm going to show you, the oldest male is about 55 currently. So he was estimated to be 25 years of age when they first started working at the site. And that may be off by 4 or 5 years, but it can't be off by much more than that because he looked old at the time. And chimpanzees reach their full adulthood, males at about 15. That's when they start to become a part of the dominance hierarchy. So the age estimates should be fairly good. All right, so just to start with the basic life history trade-offs that you're all familiar with, I want to think about the idea that we've really started to understand that within this ability to shunt energy toward reproduction, that in males at least you get this strong trade-off between mating effort and parenting effort, and that really what the steroid hormone testosterone is doing is supporting mating effort at the expense of parenting effort. And that, in fact, you can see most of the differences between males and females. Sexually dimorphic traits are driven by testosterone, and most of the differences we think of between males and females are that males invest more in mating effort. But you can think about the classic sort of things you think of as being testosterone driven. Some of them just involve the basic mating mechanics, the general differentiation and so forth, and sperm production. But it's also underlying the sexually dimorphic muscle that males use to compete with other males. It's having something to do with libido or interest in mating. And it's also facilitating aggression, but particularly in reproductive contexts, not just any aggression. In a lot of the literature, there's this clear connection between testosterone and aggression only when you put it in a reproductive framework. And of course, interest in status, interest in dominating others, which in most species is going to be connected in some way to aggression. So the point is just that testosterone really looks like it's driving mating effort. And a lot of this has been put into the framework of the challenge hypothesis. So this was worked out by John Wingfield and colleagues, and I think now there are upwards of 84 or more species of birds that they've tested this with. And it's a very good explanatory framework for what happens in birds. And the point of this is just that most of the variation that you see in testosterone in the wild is going to be having to do with aggression in reproductive contexts. So what you see in birds is that they'll go from a non-breeding baseline of testosterone to a breeding baseline, and they're just elevating their testosterone levels a little bit, and there appears to be a kind of threshold effect. You need a certain amount of testosterone to start producing sperm, to start getting libido going. But then from that breeding baseline up to this maximum physiological level, which is just set by the number of testosterone receptors you have, you see a lot of variation in different bird species. And what you find is that it's clearly tied to this aggression and this mating effort, parenting effort trade-off. So during parts of the breeding season when males are competing over females, they'll have really high levels of testosterone up here. And if they're a species that doesn't invest much in paternal care or provisioning, they'll just keep that high throughout the breeding season as they're fighting with other males trying to monopolize females. If, on the other hand, you're a species that once you have a clutch of eggs, you're out provisioning them every day, you find that levels drop back down here until you get to the next clutch and so forth. And there's even nice experimental data where you can take a normally monogamous bird, put a testosterone implant in it, and the males will go around trying to monopolize a bigger territory with lots of females. So the basic point here is and this sometimes gets lost in the challenge hypothesis has become sort of trendy recently in looking at the human males, and it's sometimes just put in a framework. Anytime you see any sort of competition, it must be the challenge hypothesis. But really, there is a strong expectation here that it should only be competition that's in a reproductive kind of context. And the idea is that most of the variation in testosterone you see is going to be to do with that rather than just with changes in reproductive physiology. So to give you an example of that, you can take two primate species that both, so the rhesus macaque here and the marikis, that are both seasonal breeders, both live in multi-male, multi-female kinds of social systems, both have intense sperm competition, but one has aggressive mating competition and one doesn't. And in the one that has the aggressive mating competition is where you see the big increases in testosterone in the breeding season. In marikis, you don't see that. So this is the idea that it's the physiology of sperm production and so forth is not as sensitive to these fluctuations. There's also been some effort to look at sort of long-term fluctuations in testosterone and how that maps onto male reproductive effort across the life course. And this is just one nice study that was done by Jacinta and Wiener and colleagues on a number of different baboon species. And it's just showing you the trajectory of dominance rank in males and then how that maps onto the testosterone trajectories. You can see in, let's see, these were the Chacma baboons here. There was actually the increase in testosterone preceded the peak dominance rank. Slightly different pattern here, but you can see in general that it's the period when they're contesting for rank that you get the higher testosterone. And I'll show you some stuff on chimps in a minute. But just to start with this challenge hypothesis, it makes some fairly specific predictions about what you would expect to see in chimpanzees. So the first thing, male chimps should show these increased aggression and along with that increased T levels during periods when they're fighting over estrus females. And then we want to know, is that increase actually related to the aggression or is it just a byproduct of sexual behavior or the physiology somehow? And we've looked at that in a number of studies at Kanyawara. I just wanted to show this briefly because we have some new data which just nicely support everything we've said before. But it's clear that male chimpanzees are much more aggressive when there are estrus females around, and particularly when there are estrus parus females around. So the mothers are much more attractive than the young nulliparous females, and the males compete over them. So not as big an effect here for the charging displays, but for real contact aggression and the chases and attacks and so forth, you get an increase, which increases especially during the periovulatory period, so the window right around ovulation when conception can actually occur, you get a further increase in aggression. And again, this is really something that you see with the parus females, not so much the nulliparous females. This is just to show you some data on the fact that male chimpanzees are much more interested in these older females than they are in the younger ones. These are just 4 different measures of that. This is the percentage of copulations that the male initiates by approaching the female versus the female approaching the male. And you can see the nulliparous females, in order to get males interested, have to approach the male, whereas the older mothers can just wait for the males to come to them. This is the average rank of the copulation partner. You see the low-ranking males end up mating with the nulliparous females. The high-ranking males prefer the older ones. This is just a measure of if you look at the average party size for a given month, how does that compare to a party with an estrus female in it? How many additional males would you get in a party with an estrus female? And you can see the effect is greater for the older females. And then just the rate of aggression you see is more for the older parus females. So this allows us a pretty straightforward test of the challenge hypothesis, because it turns out that the males are copulating at the same rates with the nulliparous females as they are the parus females. So the sexual behavior hasn't changed, but what's really different is the competition. The males with the parus females are trying to keep other males away. They're trying to monopolize them, whereas the nulliparous females, they're still mating with them, but they're not interested in monopolizing them. So you get the sex with and without the aggression. And if you compare the testosterone levels, so this is a recent analysis from just data from 2005 to 2010. You can see there's no difference at all in the testosterone levels when there are nulliparous swollen females around and no swollen females in the group. But then when you put the parus females in, you get about twice as much testosterone on average. And I neglected the statistics here, but these are not different from each other, and this is different from both. So that's the short-term stuff, and this is really just a replication with different males at our site of something I did in my dissertation. More recently, we've wanted to look at this long-term pattern, like the baboon pattern I just showed you. So the question here is just during the periods of the lifespan when males are really competing for mating opportunities, competing for dominance rank, do you see testosterone levels and levels of sexually dimorphic muscle, too, following along that same trajectory? So we've recently looked at a long-term data set. So this is based on, almost 5,000 samples. And all I've done here to make it real clear is for each age, I've just averaged the testosterone levels from the year from all the males that were seen at that age. So each of these points represents between 1 and 8 males. And you can see the trajectory of testosterone is fairly flat until about age 10, when they suddenly have this big increase and they reach adult levels of testosterone about 15, which is right when we see them start to enter the adult male dominance hierarchy and start competing with other males. And then nicely at the other end, you can see these declines in testosterone with the older males. So one of the things we haven't looked at in a lot of detail yet is what's going on. I mean, what happens when you're sort of a past prime male who can't really compete anymore? What kind of a strategy do you take? But the trajectory here is a little different than what you see in captivity. This really isn't that surprising, but if you look at captive chimps, they start to increase their testosterone levels about 6 to, and it really by 8 years it increases, and by 10 they've reached their adult levels of testosterone. So it's consistent with what we know about captive female chimps. They start cycling earlier, they reproduce earlier, but it's nice to see the same effect of energy on male development and testosterone levels. All right, so this graph could be more elegant, but I just picked the same 5 years of data that were represented by the challenge hypothesis data I showed you later. So this is the same kind of graph, just showing you at each age the average dominance rank of individuals of that age. And you can see that the real peak here, all of the alpha males at Kanyawara have been between 20 and 30. So 30 years of age is about the expiration date for an alpha male. Like they can make it up there by 19 or 20, and then the one who has served the longest was about 10 years. But we have 4 alphas now who, right about the time they hit 30 is when they just couldn't compete anymore. And so, you can see some are able to maintain a high rank longer than that, but in terms of being the number one, that's about it. But you can see the pattern looks a lot like the testosterone. I mean, a lot messier, but the same sort of peak right around age 30 or just after. So let's see. So here, what I did is I just showed you the average of all the males at each age. This is a messier scatter, just showing you the individual data points. So each of those is one male. His average testosterone levels for a year when he was that age, and that's just a smoother to give you a general picture. But the reason I'm showing you this is just because you can, if you look at the males who maintain high rank when they get into these older ages, so the green dots are the higher ranking individuals, and the red dots are the lower ones. You can see the green dots tend to be up here. So even these elderly males, if they're the males who manage to hang on to high rank, they're the ones who are maintaining high testosterone into those older ages, and the ones who don't are the ones who are lower. So what about muscle mass? We'd like to know we know one of the major functions of testosterone is to promote the growth of sexually dimorphic muscle that males use in competition. But it's obviously very difficult to measure this in the wild. We can't weigh our chimps like some sites have been able to do, and we certainly can't do sort of measures of body composition. But we found this really kind of neat method recently. So it turns out for years people have used measures of urinary creatinine to correct their hormone levels. So when you go out and you collect a urine sample from a primate in the wild, you don't know whether the variation you see in that sample is because some individuals were drinking more water than others, or these kinds of issues. So what everyone does is they correct for creatinine. So creatinine is produced by skeletal muscle at a relatively constant rate. So you can sort of correct for dilution effects and time effects by saying how much testosterone was produced per unit of creatinine. And of course, this only works if you're comparing individuals that are sort of roughly the same lean muscle mass. If you're trying to compare infants with adults, you can run into trouble because the adults are going to be producing more creatinine. And this is just showing you in humans, there's this nice expected relationship between 24-hour urine sampling, which you can get in humans and not chimps, and how much creatinine is produced. So each kilogram of lean muscle is in human men producing 23 milligrams of creatinine. So we recently decided, well, why not try to come up with a creatinine index to get at muscle mass differences in chimps? And the way we did this is just by, in any given urine sample, looking at how much creatinine there was compared to the specific gravity of the sample. So how much creatinine compared to all the other things in the sample. And for each individual, you need a lot of data to do this, but since we had 12,000 samples from all these chimps, you can, the data I'll be showing you in the next slides are actually the slopes from the relationship between creatinine and specific gravity. And you can see just the plots here. So infants have much shallower slope than juveniles and subadult females and adult females, up to the adult males that have the most muscle mass. So the idea is if you can get enough samples from an individual and calculate a slope between these things, then at any given specific gravity, males who have, or individuals who have more muscle should be producing more creatinine. So here's a nice finding where so each of these points is an individual male for a year, and it's the slope of this relationship between creatinines and specific gravity. And that's just plotted against the same male's average testosterone levels for the year. So you can see we have what looks like a nice measure of muscle mass. There's this direct relationship between testosterone and creatinine. Now this doesn't look particularly strong, but this was just all of the data. If you actually restrict it to the slopes that had at least 30 samples to make them, you can get a better fit. So some of those real outliers where you were calculating a slope maybe from 4 or 5 samples, but we used a cutoff of 30 to get a fairly robust measure. And here you see the same kind of graph that I showed you before. For each age, I've just calculated the average of the slopes of creatinine for specific gravity for the males at that age. And you can see it's following pretty much the same trajectory, where the males right around this age of 30 are reaching a peak. Maybe not quite as, some of the older males had fewer samples, so it was harder to get all the really elderly individuals. But and then you can just do the same scatter plot I showed you before, and show that you don't get the same effect for muscle mass with the high and low rankers that we saw with the testosterone. So some high-ranking males have less muscle mass than others. And this was expected. We already know from chimpanzees that it's not always the biggest male who's the alpha male. It's like this, several years in here are driven by a small male who was the alpha, who's just particularly good at manipulating coalitions. But we're now working with these data to try to see sort of more sensitively what's going on with these old males. Is there, what are they doing when they become old? Is it that you pay a cost in investing in muscle mass and male-male aggression early on, and that there's no high-ranking males out here because all the ones are dead? Or is there just a shift to a strategy that's not as risky? But as just a brief aside, I'd like to suggest that there's a real difference here with testosterone and aspects of male physiology in chimpanzees from what you see in humans. So in humans, there's lots of good evidence that behavioral aspects of mating effort are tied to testosterone levels. So there's a huge number of studies now like these ones, including this one I did with the Hadza in Tanzania, showing that men who are invested in a long-term relationship, and particularly men who have children in those relationships, have lower testosterone levels than men who are unpaired or married but without the children. This is a sample from the Harvard Business School. This is just a sample of Boston men. These are Hadza men. These are AM samples and PM samples from the Hadza. But there's all kinds of data now and there's including longitudinal data. So recently Chris Kuzawa and his colleagues published a nice paper showing that it's not just because high T men don't get married and have children. They actually showed that the high T men at the beginning of their study, which is a longitudinal study of several hundred men in the Philippines, the high T men were more likely to get married. But once they got married and had kids, they were downregulating testosterone. And there's a whole nice literature in evolutionary psychology showing that men with high testosterone are more interested in extra pair mating, novel partners, all kinds of things, and that they're less interested in infants and children, and that women can even see pictures of such men and accurately gauge their interest in infants and children. But if you look at the connection between musculature and strength and testosterone in humans, very distinct from these behavioral patterns, it's not nearly as strong. And in fact, any of these studies that have looked even across individuals or even within individuals at different times, there's a very, very weak or non-existent relationship between testosterone and muscle mass in humans. I mean, it's sometimes there and you can pick it up, but it's amazing how rarely. Here's an example of that disconnect. These are some data that a grad student of mine, Louis Alvarado, collected in Poland. He picked this population in Poland just because they were doing heavy physical farm labor. He wanted men who were actually working. And you can see he's just broken it down into age categories here, but for these measures of body fat or lean muscle mass, so arm circumference adjusted for fat, and here's some measures of chest strength and grip strength. These farmers who are out working every day maintain their strength up into these older age categories, up until they're 60. But their testosterone levels follow the kind of profiles you see in other populations where men start to decline early in their mid-20s or 30s and just go down from there. So, and this is a common feature. He's also looked between seasons. So in the season when men are working really hard versus in the winter when they're sitting around not doing much. And their testosterone levels either don't change or they're actually higher when they're sitting around, yet their measures of strength and fat change dramatically. They're much weaker during the winter season. They have more fat then, but if anything, they have higher testosterone levels. So there's a real disconnect. And I think one of the reasons, and this has been neglected in a lot of the literature, which sort of assumes that these things all go together, is that if you think about the sexual division of labor and what men are doing, I mean, it makes sense to downregulate the behavioral aspects of testosterone if you're investing in a wife and children and you're going out and working and provisioning. But it doesn't make much sense to decrease your investment in musculature or sexually dimorphic musculature just the time when you're expected to do the most work. So, I mean, we know from the Hadza and other studies that men with young children are out doing a lot more foraging than men who don't have such dependence. So it really looks like there is this slight disconnect, or men are not as sensitive to these decreases in testosterone when it comes to their strength and workloads, whereas we see the behavioral picture looking very similar to other species. In chimps, on the other hand, I think there's a pretty direct connection between investment in the sexually dimorphic muscle, because pretty much what they use it for is to fight with other males over access to females. That we have students who are looking into this more, but I think it's an interesting area of difference just because it's one of the few species, primate species, where men are using sexually dimorphic muscle not just to fight with other men, but to provision their families. Okay, I want to think a little bit about other aspects of male mating competition. And in particular, some of the work we've done about male aggression being used against females as a form of mating effort. So for anyone not familiar with chimpanzees, the kind of aggression you see by males against females is very striking compared to what you have in a lot of other species. So it's not uncommon in some primate species to have sort of a kick or a slap in passing. But with chimpanzees, you can get these really extended, brutal attacks where males are lifting females up and slamming them onto the ground and kicking them and beating them and sometimes wounding them quite seriously. And if you look at the skeletal collections of chimpanzees even, there's a nice study showing that females often have these blunt trauma wounds on the back of their head, presumably because they're sort of crouching down while they're being attacked, whereas males tend to have them on the face because they're fighting face to face. But anyway, there's all kinds of aspects where male chimpanzees are not very nice. And it's been suggested for some time that this might function as sexual coercion. And here's the classic definition by Barb Smuts and father about how do you get sexual coercion. So it's aggression that's directed at a female that does one of two things. And often this distinction is lost in the literature. It's either what I call direct coercion, where you're using force to make a female who doesn't want to mate with you mate, or what I call indirect coercion, where you're actually using aggression to prevent a female from mating with other males. And she might want to mate with you, but you want her to only mate with you. So there's a distinction here. Obviously, humans have both of these patterns. And then there's this key element here. There has to be a cost to the female. Because you might imagine a situation where females don't like aggression necessarily, but say they use aggression against them to somehow judge the quality of a male. There might actually be some benefit to the female aggression, but for it to actually be coercive, you want to show that there's some sort of a cost imposed on the female. So we've looked at a lot of data on male aggression against females from Kanyawara, and it's clear that there's a link between aggression and sexual competition, sexual behavior. So this is just showing you again for the Paris females. This isn't true for the nulliparous females. For the Paris females, they receive much higher rates of aggression from males when they're in estrus, when they have these swellings and they're near ovulation. And this is controlling for time spent together and all those other kinds of things. You can see these are just individual females. And you see the dramatic increase in aggression when they're actually swelling. This is showing you the corollary of that males are actually mating more with the females that they're aggressive toward. So the axis here is now males. Each of these is a male. And what you're seeing with the white bar is the average copulation rate of that male with all the females who he was least aggressive toward. So the females he showed less than the mean amount of a female aggression for that male toward. And then the black bars are showing you the copulation rates with the females that he was most aggressive toward. So you can see they're copulating more with the females that they're beating up on. This doesn't really give much insight into the mechanism. Are they overcoming female reluctance, or are they just sort of preventing females from mating with other males and thereby overrepresenting their own copulations? But it clearly is consistent with that idea of sexual coercion. What about a cost? Well, we've used stress hormone levels in females to try to get at this picture. And here's just showing you that the stress hormones go along with the behavior. So with the Paris females, you get these big increases in aggression when they're in estrus. With the nulliparous ones, you don't. And if you just look at cortisol excretion in the two groups, you see the Paris females have much higher levels of cortisol. within the Paris females, they have much higher cortisol levels when they're fully swollen than during periods when they're not cycling. So this was just compared with a period of lactation. And in fact, when we recently put together a much larger data set and just did some mixed models to see what were the major predictors of cortisol levels in females. There's all kinds of things that can make your cortisol levels go up, not having enough food, aggression and so forth. But you can see the strongest effect is aggression, female aggression some, but the real predictor in this particular data set. And what we did for this is we took the average amount of aggression that the average cortisol levels of females by month, and then looked at variables per month that explained those. And the amount of aggression received from males in a month was a strong predictor of a female's cortisol levels. So is this really coercion? Well, we've been interested in this idea, and there have been a couple of studies of chimpanzees, including these ones by Becky Stump and Christoph Bosch, suggesting that female chimpanzees, there's a strong element of female choice in their mating. And the measures they've used are female proceptivity and female resistance. So the idea is if you can look to see, does a female approach a male for a copulation, or does the male approach the female? So you can see, you don't have to be an ethologist to see what this male wants. They're pretty straightforward in their behavior. And what she did is she scored females in different parts of the cycle on their proceptive behaviors and then their resistance behaviors, which was just whether you ignore or avoid a male when he solicits you in this manner. And what they found is that some males were consistently approached right around ovulation, and some males were consistently rejected right around ovulation. And they argued that this was a strong form of female choice, that females might end up mating with all the males, but the ones they really want to mate with, they're mating more within this window around ovulation, and the ones they don't like, they're mating less with. And the problem with these kinds of studies is that none of them really grappled with the problem of sexual coercion. So the problem is if a male comes up and solicits you in this manner and you choose to ignore or avoid him, does it really mean you don't like him, that he's a non-preferred mating partner, that you don't like his genes for some reason? Or is it because there's a male over here who's watching you, and if you mate with him, he's going to beat you up? And if you rush to show proceptive behavior toward a male, is it because he's really attractive and has some indicators of high fitness? Or is it because you have experience with this male and you know that if you don't do this when you're cycling, he's going to be aggressive? So we looked at this a little further in our data and found actually that we limited this analysis just to copulations that were initiated by the female during the periovulatory period, so during the window of highest fecundity. And so what you see now is individual females, and it's showing you the number of times they initiated copulations during this period with males who were the least aggressive toward them and males who were the most aggressive toward them. And you can see for 4 of those females, they never initiated copulations in this period with the males who were the least aggressive toward them. So it's striking. It sort of suggests that you need to be careful in assuming that just because a female is showing proceptive behavior, it's an honest indicator of preference. The one last thing I'll say on this is you might want to ask, why would female chimpanzees want to bias paternity? So in the human literature, this is a big thing. And I think some of the confusion often is just that we know female humans have very strong mating preferences, so we just assume female chimpanzees ought to as well. But you can ask yourself, what would be the point of biasing paternity? Well, in a lot of primates, the answer to this has been infanticide protection. The idea is that you should bias paternity toward a high-ranking male because he'll protect any offspring he produces from being killed by other males. The problem with trying to apply this to chimpanzees is that they have this fission-fusion social system where whoever fathers your offspring is likely not to be with you most of the time, so you'll always be vulnerable. So there's a question of whether that really explains much. Most of the other models of paternity bias fall under these sort of good genes kind of explanations, which tend to be either a good genes model where all females are expected to choose some male who's really excellent, or occasionally you see these sort of compatibility models where females can maintain their individual preferences. We've tried to look at this a little bit in the Kanyawara data just by looking at who females are being perceptive toward. You can find particular males that females are often approaching and then ones that they aren't. And you can see this is the alpha male Emoso, who is actually a preferred male. He's approached at very high rates by all the females in the group. And then all the other males differ, except the lowest ranking male is no one likes him. But there's this sort of heartwarming finding that there's someone for everyone. At least every male except poor Stocky there has at least one female who approaches him, he's a preferred male. And of course the problem here is that the alpha male was among the most aggressive toward all these females. So you can't really escape this problem with aggression. But anyway, we tried to find what would some predictions of these models be for chimpanzees. And one of them should just be that under any kind of paternity concentration model, if a female is actively trying to concentrate paternity in a male, whether because he has good genes or some other thing, then that male's interest should be aligned with the female's during the periovulatory period. So here's the alpha male. These females should be trying to concentrate paternity in him, and he wants them to do that. So what you should find is during this period there should be less conflict between them. So we just looked at rates of aggression in these pairs during the pre-pop days of maximal swelling, so the period when they're mating but they're not likely to get pregnant, versus the periovulatory period when they're most likely to get pregnant. And you see there was no difference with any of these. If you looked among the males who were approached generally in those dyads, or the alpha male with all the female dyads just because he was an approached male for all of them, or within the non-approached males even, there was no difference. So there's no hint here of a lessening of this conflict of interest. So an alternative hypothesis that we proposed is just that maybe female choice is a little different in chimpanzees than it is in humans. So we know that infanticide is a big risk for female chimpanzees. And if you start with the assumption that females really want to mate with all the males in their group and convince them that they might have a chance of being the father to avoid infanticide attempts, then maybe this kind of preference that you expect female humans to show doesn't make sense for a chimp. And that really the more a female, if a male is really successful in getting a female to concentrate all of her copulations in him, it's very dangerous for her and her offspring. That there's really a strong motivation for a female to mate with lots of different males. And maybe these apparent preferences you seek are just the result of male coercion on top of a system where females are basically their main concern is to avoid infanticide. So it's not to say that females don't have preferences, that females don't have agency. It's just that their agency is directed toward a different goal than human females. It's to make sure that their infants are protected by giving all the males a sense of shared paternity. So one thing we looked at, this was actually in response to comments from Joan on this manuscript, was we looked to see what was the rate of female-initiated copulations toward each of these males when the alpha male was gone, when he was off in a different party somewhere, versus when he was present. And you can see as soon as the alpha male leaves, all of these males show increased rates of mating with these females, some of them more than others. There's maybe still room for some preference in here. And then this is the reverse. This is male proceptivity toward a female in the absence of the alpha male. And you can see they all show quite low rates of compared to when the alpha's gone. So there's clearly something about these aggressive males that seems to be constraining a bit. Okay, I think the time, real quick, I'll just show you a few things on costs, because there's some kind of fun data that have just come out recently. So we're just interested in the idea of, what's constraining high-ranking males from doing this. So there's one idea that there's a life history cost. If there's only so long you can maintain high rank when rank is maintained through aggression before it starts to wear on you. But even within ranks, you see things like this. These are data from Gombe, where they've looked at paternities, and this is the sort of predicted paternity distribution from a straight priority of access model. And this is the actual paternity. So the alpha males are doing well. In all chimp populations, alpha males do better than other males. But at Gombe in particular, the lower ranking males are doing a lot better than you would expect them to. So why can't the alpha males successfully constrain things? And one of the things we've just been looking at is what are all the difficulties? What are the costs of being an alpha male? Some of this we've looked at just in terms of stress hormones. So we found that consistently in different study populations that it's the high-ranking males that have the highest levels of stress hormones. That's a reversal of the expectation in some species. Here's some data from Gombe showing the same thing. But it's not exactly clear what's going on. People often want to pretend that cortisol is sort of a measure of psychosocial stress, whereas in fact it's a metabolic hormone that underlies physiological stress. So energetic stressors can cause cortisol levels to go up as well. So one idea is just that if you're a high-ranking male, it's not so much that you're sort of psychosocially stressed, because, I mean, in fact, the high-ranking males win most of the aggressive encounters that they're involved in, and they probably have a greater degree of predictability and control over their lives than these low-ranking males most of the time. Yet they seem to maintain high cortisol levels, and one idea is that this is just because there are considerable energetic investment that goes along with being a high-ranking male and maintaining access to females. So the next few slides I'm going to show you are some data from Alexander Georgiev, who I just came back from his thesis defense. He was looking in detail at what are the costs of mating for male chimpanzees in terms of various measures that would affect energetics. So just quickly, he showed that male feeding time declines when there's a paris estrus females around, but not for nulliparous, or not statistically significantly there. And so this is daily percentage of time spent feeding. And then the duration of feeding bouts is actually shorter. So in the presence of paris estrus females, males terminate their feeding bouts quicker than usual. And if you add all this up, what it actually means is when there's a paris estrus female around, males lose about an hour of feeding time a day on average. And it seems to be because there's a cost to monitoring an estrus female and fighting with other males over her. It interferes with getting energy. Now you might ask, well, okay, maybe they're feeding less in terms of time, but maybe they're choosing higher quality items and somehow making up for it. Well, that doesn't seem to be the case. These are more of the Georgiev data. He shows that the paris females, you can see if anything, they're spending less time focusing on ripe fruit, which is the really good high-quality item in the chimpanzee diet, as opposed to pith and leaves and so forth. And then if you just look at the percentage of the diet that's ripe fruit, it's not increasing in the presence of paris females. And then if you look at what's eating into their time, you can look at rates of male aggression and rates of male copulation, and just see that the more you're doing of these things, the less time you spend feeding. So does this have effects? Here's just a graph showing the overall effect with rank. So the high-ranking males are feeding a little bit less. And I showed you similar data before, but these are just the aggression data from this particular study. You can see this is the alpha male here, and you can see during the squares here are the periods when there's a paris estrus female. You can see that's where you get the much stronger association between rank and aggression. In all cases, the alpha is fairly aggressive, but these other periods are ones where there aren't these paris estrus females around. But does this actually have effects in terms of energy balance? Well, we've recently been using another biomarker, C-peptide, which is a byproduct of insulin. So for every molecule of insulin that's produced in your pancreas, you get this little C-peptide unit cleaved out, and it's cleared into urine. So it's very nice because there's a one-to-one relationship between the amount of C-peptide in your urine and the amount of C-peptide that's been circulating. And it's something that's very easy to measure. So consistently high levels of C-peptide are associated with in positive energy balance, so taking in more calories than you expend, and then lower levels as you get to negative energy balance. And there's some nice experimental studies in captive chimps with feeding and refeeding experiments or feeding restriction experiments. But you can start to look at this and find that C-peptide levels are much lower on these days when they're feeding less and expending more energy and aggression and so forth. You can find that, here's the aggression rates and C-peptide. And then this is, again, Georgiev data showing that the high-ranking males actually have lower levels of C-peptide, which might not have been, people might have expected, okay, the high-ranking males are doing all this work, but maybe they're somehow making up for it by having better access to resources or something. But all the physiological indicators indicate that the males are paying a real cost to engaging in all these. They're not getting as much food. And this was just in his particular one-year study. This is a data set that we published over a much longer period, but showing the same thing. The high-ranking males down here having lower chronic levels of C-peptide. Again, this is just the graph I showed you before of testosterone with age, and what we really like to do now is look into more detail about what's going on with these declines out here and see if we can measure these costs and whether they have impacts on males at these later age. Because there's obviously sort of two kinds of things that could be going on at least. One is that if you expend all this energy being high-ranking, that you just can't do that for very long, and you show signs of morbidity and you die. We do have some data like that showing that alpha males are more likely to show signs of diarrhea or coughing or all these other kinds of crude measures we can get of health. The other alternative, of course, is that you only invest in that strategy of being high-ranking if you're somehow in phenotypically very good shape, and that there's some sort of self-selection there where it's only really high-quality animals that are able to do that, and we won't actually see that pattern of senescence. And it's a difficult kind of thing to tease apart, but we want to look at things like males who just maintained high rank throughout their life, males who stayed low-ranking throughout their life, and then maybe ones who tried to be high-ranking but failed, and see if we can get any kind of purchase on those problems. But that's just some of the recent data we have on costs of high ranking. And this is something that's showing up in more and more studies. I mean, I know in the baboons studies that there was the cortisol was much more intense for the alpha male, and then the beta and the males who were just below that in the hierarchy didn't seem to have such an effect. They were actually lower than in cortisol than the really low-ranking males. But with the chimps, the two are, it's fairly consistent that the 2 or 3 top-ranking animals have higher stress levels and these other indices of physiological costs to maintaining that kind of rank. So there's everyone here.